Okay, so this next lecture is about protein metabolism during different states, including fasting, fed, stress, etc. And so what happens to your proteins and more importantly, what happens to nitrogens. So we'll talk about different organs and how they use amino acids or how they release amino acids, and how these are going to be different depending on the metabolism state of the individual. So we already know that proteins during the post-absorptive state and fasting state, your proteins in your muscles are going to be broken down and you're going to release amino acids. Your muscle cells will also release glutamine, and glutamine has a few important things going on. Glutamine is also going to provide carbon sources for your kidney and for your gut to make energy. So I'm talking about glutamine right now. And we can also put the gut over here. So your glutamine is going to also provide nitrogen, which is important for the kidney. And the nitrogen is going to be excreted in the urine. And this is going to be a buffer. Now, removing protons during oxidation of fuels by this process of buffering the urine with nitrogen is going to be important in maintaining the body's pH. Oh, darn. And this is important especially in periods of acidosis. You don't want to use bicarbonate to buffer the body during periods of acidosis because you need bicarbonate for the urea cycle. Also, in rapidly dividing cells, so we have here our macrophages, lymphocytes, fibroblasts, glutamine is going to be a fuel as well as a nitrogen donor for the building up of proteins. It's fuel. Write that on a little bit better. Okay, so now in a hypercatabolic state, and a hypercatabolic state is a state in which you have negative nitrogen balance. So it is a state of stress. And it could be in periods of sepsis, trauma, some sort of injury, or burns. And you're going to have a need for more nitrogen, so you're in a negative nitrogen balance. Okay, so what's going to happen in a hypercatabolic state is that cortisol is going to be the major hormonal mediator of mobilizing nitrogen sources. Okay, so where is nitrogen going to come from in your body if you have a negative nitrogen balance? Well, you're going to tap into your proteins, of course. So cortisol is going to stimulate proteolysis via ubiquitin. So this is the proteasome. And cortisol is also going to stimulate release of amino acids, especially from muscle cells. And cortisol is also going to stimulate the synthesis of glutamine synthetase. So an increase of protein degradation in the muscle cells, especially your skeletal muscle cells, increases the synthesis of glutamine and other amino acids for cell division. This also helps to increase the nitrogen pool for protein synthesis in cells involved in immune response and wound healing. Oops. Right. So in periods of hypercatabolic states, you get an increased release of your glucocorticoids from your adrenal cortex. And these, of course, are also going to help to stimulate proteolysis as well. So what's really important is the maintenance of a free amino acid pool in the blood. And this is because you need to do all of these things. You need to synthesize new proteins. It makes sense. Cells that are rapidly dividing are going to need to make purines and pyrimidines. Your red blood cells need heme. Your brain, your central nervous system, your peripheral nervous system, they need to make neurotransmitters, hormones, etc. What comes in is going to be your diet. So your dietary protein, of course, is going to influence your pool of amino acids, as well as the breakdown of proteins already inside of your body. Now, when you need to use the carbons in your pool of amino acids for ATP, then you're going to, of course, need to make or deal with your nitrogen. So here you're making ATP. You're going to need to deal with your nitrogen. You're going to form urea. You're also going to buffer the urine with your nitrogen ion. And so all of this is very important just in basic homeostasis. Now there is during the post-absorbent state or a fasting state, your protein synthesis is going to continue. Your degradation of your proteins are also going to occur in your skeletal muscles, and your skeletal muscles are what's going to be releasing your amino acids. So skeletal muscles in a overnight fast, for example, will break down your branched-chain amino acids, and this is going to use they're going to use your carbon sources for energy, and then they're going to release your nitrogen in the form of glutamine. So your skeletal muscles will transfer the nitrogens to either glutamate to make glutamine, or they can also transfer it to alanine, of course. Alanine's down here, and we have glutamine. Now, you're also going to, of course, in a post-absorptive state, have a decrease of insulin. And this is going to increase proteolysis. Just in general, because again, you're mobilizing fuels and fuel sources. And so overnight fasts are going to lead to a net increase in the release of amino acids. Okay, so a little bit about the liver. So liver, of course, is going to be important in taking and removing nitrogen during a fasting period. So liver is going to be taking up alanine and other amino acids on here. And liver is also going to be taking those amino acids and oxidizing their carbon skeletals primarily to make glucose. And then your nitrogen that is going to be coming from these amino acids is going to be donated into the urea cycle and generate urea. Glucagon and other glucocorticoids can stimulate the uptake of amino acids into the liver, and they also are going to increase gluconeogenesis and ureagenesis. So glucagon or epi, for example, will have the same effect on your liver cells. Now, when we go to other tissues during fasting, a few things happen as well. Now you have glutamine, which we generated in our skeletal muscle already because they were breaking down branched chain amino acids. And also your lungs and brains are also going to use branched chain amino acids. So your brain will pick up your branched chain amino acids and use them as well. The kidney and gut, these are organs that have rapid turnover rates, so they need to regenerate cells. So they are very metabolically active even during an overnight fast, for example. They are going to take up a lot of the glutamine. So you have your kidney here. They're going to pick up glutamine as well as your gut. The gut is right here. And the gut will pick up glutamine. Let me make it a different color. So glutamine is going to be picked up by your gut and kidney primarily because they need those carbons for energy. And then the nitrogen is especially important, of course, in buffering the urine, as we've just been talking about. Alanine is going to take unused nitrogen back to your liver. So alanine's down here. Oops. And that's going to go to the liver. The brain is going to be glucose dependent. However, they can also use the branched chain amino acids for energy. And branched chain amino acids going to your brain are also going to be a source of nitrogen to make neurotransmitters. Okay, and so this is just kind of how your movement of nitrogen occurs during a fasting period. Now, there are a few principles that are going to influence this amino acid flux between tissues, if you call it that, and it's going to be affected by the exchange of fuels. I should put this, hang on. So if you go back into the fed state, for example, or if you have some sort of disease state, sort of a metabolic acidosis, for example, or if you're going to have some sort of trauma, as we've talked about. And this can be surgical trauma, it can be an injury trauma, it can be a burn, sepsis, wound healing, so after trauma. And a lot of it has to rely on the need for nitrogen. So if you need nitrogen in order to repair your cells, your tissues, you're going to have to tap into your proteins in order to release those nitrogen and amino acids in order to repair tissues that are damaged, for example. Okay, so we have here also what's going to be influencing our amino acid flux is going to be ammonia. Ammonia, of course, is toxic, has to be taken care of. Also, another influencer is glutamine. So how much glutamine is going to be in the blood? So glutamine is going to have many roles. One, the nitrogen is going to be a buffer in the urine, and it buffers the pH of the urine, of course. It's also going to be a fuel source for metabolic active tissues. And this can be the gut, kidney, immune system, for example. It is also going to be important in the brain. So in the brain, it's going to remove ammonia, and it's also important in transporting glutamate between different types of cells in the brain. And remember, glutamate is an excitatory neurotransmitter that's important in communication in the brain. During acidosis, the kidney is going to pick up most of the glutamine. And that is so it can buffer against the acidosis. And so it's going to be the favored site during periods of acidosis at the expense of other tissues. During sepsis, in the absence of acidosis, the immune system is going to be the preferred site of glutamine uptake. Now, another important regulator of amino acid flux between tissues is the branched chain amino acids. They are going to form a significant composition of the average protein. They can also be converted to TCA intermediates, and they are also a major precursor for glutamine. Oops, sorry. And then there are also amino acids that are going to be important for gluconeogenesis, of course. And so your liver has to maintain blood glucose, especially in a post-absorptive state. And so it's going to use the carbons of amino acids in order to do this. Now, In the kidneys, so one thing to keep in mind that all tissues in your body, regardless of where it is, are going to need amino acids for protein synthesis, right? So let's take a side note. All cells need amino acids, except for your red blood cells, for protein synthesis. Right? And so what this means, and so therefore, all cells have the ability to turn over proteins. And so we talked about how this is done in the past. We've talked about how this can be done at the micro level with lysosomes. It can be done by proteasomes. It can also be done at the macro level. You can do apoptosis. Your cells can do autophagy, etc. So this is happening in all cells. Now there are some organs in which this is going to be very important. So one of the primary roles of the nitrogen and amino acids is to provide that ammonia to buffer in the urine. And so that's where it comes in with the kidney. So the kidney is going to be picking up glutamine. Importantly, this helps it to release bicarbonate. So it's going to be able to buffer the pH of the urine using the nitrogen ion rather than bicarbonate. Especially important in periods of acidosis to spare bicarbonate from being used as a buffering source. Now, of course, your kidney, just like any other cell, once it removes the nitrogen, it can form alpha-ketoglutarate, and then, of course, that can go on to do whatever it needs to do. You can break it down, you can make energy. Remember, the kidney cells also have the ability to do gluconeogenesis because the medullary cells need a constant supply of glucose. So the kidney can use alpha-ketoglutarate to form glucose, and this will go to the medullary cells. And so that's what your kidney is doing. The rate of the amount of glutamine uptake is going to be dependent on the blood pH. If you are making more urea, then you are going to have to excrete that, and so you're going to have to buffer your urine more. In periods of metabolic acidosis, you increase the excretion of your ammonia ion several fold. And then the proton excretion from your kidney has precedence over the requirements of other tissues for actual glutamine. So in periods of acidosis, your kidney gets preference. For glutamine. And this is at the expense of other tissues. So you have to think your body is trying to counter the most important threat at the time. So acidosis is a huge threat because your enzymes cannot function at certain pHs. And so it would be like a global loss of enzyme function. And of course, the body can't function without enzymes. And so that becomes the most important thing for your body to counter. And so it uses the kidney to try to balance the acidosis using glutamine. Okay, so this is just a picture of the kidney. It's kind of going over in just a little bit more detail what we just discussed, so nothing new here. So in the kidney, you're going to pick up glutamine. You're going to get your nitrogens out, and those are going to buffer. And then in the kidney, of course, you get your alpha-ketoglutarate. Your alpha-ketoglutarate can make ATP if it wanted to. Actually, let's do this a different color to tell the story. So that can make, but remember, Your kidney also has PEP carboxykinase, and especially your cortex cells. Your medullary cells are dependent on this. So PEP carboxykinase is going to either make glucose, or of course it can go back and make pyruvate. Your kidney is also able to do a transamination reaction if it has too much of the nitrogens, and it can generate alanine, and then alanine would of course leave and go to the liver. Your kidney has the ability, of course, like all cells, to pick up fatty acids. So it can pick up fatty acids, it can go to the acetyl-CoA, etc. So the kidney has a lot going on, but one of the key things to keep in mind is it's going to be a major site of glutamine, picking up glutamine. It also has the ability to do gluconeogenesis within the kidney. The gluconeogenesis of the kidney cannot compensate your liver. So the kidney cannot maintain blood glucose level. It might be able to do it for a very short period of time, but if you lose your liver, your kidney is unable to pick up and make enough glucose for the entire body, especially your red blood cells and brain. Now, just a little bit about skeletal muscles. So skeletal muscles, of course, are going to be, remember back what we talked about. Skeletal muscles are insulin sensitive. They do not respond to glucagon. So they're very much in the fasting stage. This isn't fed. So in the fasting stage, it's very much about need. What does the skeletal muscle need? Or what kind of signal from the outside is signaling your skeletal muscle? So after a high protein meal, your insulin is going to not be as high because you don't have that high glucose, but proteins do stimulate insulin release. So we have a high protein meal. And this is going to trigger low insulin. And this will cause your skeletal muscles to take up amino acids. And this is going to lead to protein synthesis. During fasting, you're going to have that decrease. This is going to be kind of confusing here. Let me just finish this statement. Okay, now during the fasting stage, as we know that you are going to have a decrease of blood insulin levels, you're also going to increase your blood cortisol levels. And we already just talked about cortisol, and this is going to lead to a net protein degradation. And so you are going to have your carbon skeletons, and you're going to have your nitrogen. And this is primarily going to be by your branched chain amino acids, which are going to be degraded. Remember, branched chain amino acids are a major component of all of our proteins. So when we break down our proteins, we have a lot of branched chain amino acids, especially in your muscles. And so from that, you're going to get your carbon, which can become ATP, and then you have your nitrogen that is going to be packaged into glutamine. This is 2. So in your muscle cells during the fasting state, the oxidation of your branched chain amino acids is going to be very important. They are also, branched chain amino acids are going to be in other cells that can be oxidized. However, keep in mind, branched chain amino acids cannot be oxidized in the liver. So this is really a resource for other cells. Branched chain amino acid carbons are used by other cells during the fasting stage. The liver, remember, is just fine. The liver can use fatty acids, so the liver has no problem. And it's just simply because they don't have that keto dehydrogenase enzyme, so they can't break down branched chain amino acids. Now, in general, all tissues can use amino acids. They need them. They need to make proteins. And they can also break down amino acids, as I talked about before. So they can convert amino acids into intermediates of TCA cycles, for example. And a lot of this is going to be due to transamination reactions. So your cells are able to do transamination reactions in order, for example, to make alanine from pyruvate. So they can, that's a simple transamination reaction. They can also take your branched chain amino acids through transamination, make those keto acids, and through the dehydrogenase reaction form intermediates, such as acetyl-CoA. You can also do acetylacetate, etc. And so the branched chain amino acids are going to become first their keto acids. This is going to be important, as we talked about, in skeletal muscles. Let me do this. So these are branched chain amino acid oxidation. And we already talked about skeletal muscles. And it's going to be important for heart, brain, kidney. And so all of these are going to rely on your branched chain amino acid in a post-absorptive state or in the fasting state. In your liver, your branched chain amino acid transaminase and dehydrogenase levels are low in the liver. And so the liver will not use this as a source, and it won't use this as a source for carbons to make gluconeogenesis either. Branched chain amino acids are going to be oxidized by tissues that contain mitochondria. Your branched chain amino acids are converted to succinyl-CoA or acetyl-CoA, and they're always going to have the generation of NADH or FAD. So these are also energy sources. And so branched chain amino acids are going to be very important during, again, the post-absorptive state. And here's our valene down here. All right, so the major route for valene and isoleucine in skeletal muscles is going to enter the TCA cycle as succinyl-CoA and exit as a ketoglutarate. And this is going to provide the carbons necessary for glutamine formation. So here's glutamine. Now, another thing that happens is the purine nucleotide cycle. And in this case, you have ammonia from skeletal muscles, which are going to be formed from deamination of glutamate by glutamate dehydrogenase, or from the purine nucleotide cycle, which is shown here. In the purine nucleotide cycle, you're going to get deamination of AMP to IMP, and this is going to release ammonia. Now, the other way to get free ammonia in skeletal muscle is going to be deamination of glutamate. And this is going to go to alpha keto. and this is by the glutamate dehydrogenase. Right, and that can also release ammonia ion. And so these are going to, sorry, that's misspelled. These are going to then have to get repackaged up, of course, into glutamine in order to not have that toxic ammonia ion floating around. Now, branched-chain amino acids, as you see here, also feed into this. So branched-chain amino acids are going to provide the asparagine to IMP to make adenylosuccinate, which, as we know, can, through a lyase, become fumarate. And in this case, the adenylosuccinate is going to form AMP. And then you can have adenylate kinase, which can then go on to form ATP. ADP. Sorry, jumped a step there. And so this is the purine nucleotide cycle. Now, a little bit about the gut. The gut is going to pick up glutamine, and it's also going to, this is the post-prandial state, which is the fed, and then the post-absorpted, which is your fast. And in both of these cases, it's going to be able to pick up glutamine. So it picks up glutamine, and it uses glutamine as a source for alpha-ketoglutarate. And then it can go to acetyl-CoA, do the TCA cycle, etc., make energy. Your amino acids are going to be important for fuel. After you take in a protein meal, they're actually going to metabolize quite a bit of the proteins that are coming in. It's also going to be important when you're in catabolic states during fasting or some sort of trauma. The fate of glutamine carbons in the gut are to become carbon dioxide, and they also can convert their carbon skeleton to many things. They can convert it to lactate and release that, and that can be a source of carbons for your liver to make glucose and maintain blood glucose levels. Glutamate can also form citrulline and ornithine, and these can be released as well. The nitrogen can be converted to citrulline or to alanine and other compounds, and these will be taken up by the liver. You also have the carbons in alanine, which can come from glucose as well in a post-absorptive state or a post-prandial state. The oxidation of glucose to carbon dioxide is often not a major fuel pathway for the gut. So the gut primarily uses amino acids, even in the fed state. And that makes sense because the gut is going to release sugars primarily into the blood after a mixed meal. And it will keep a lot of the proteins that came in and use that as a food source. Now, in the gut also, fatty acids are not used as a significant source of fuel. So fatty acids, glucose are not significant sources. Okay, so they will primarily use proteins. So you have your proteins coming in from your diet, glutamine, aspartate, glutamate, shown here. You also are going to pick up glutamine in a post-absorptive state, as well as your branched-chain amino acids in a post-absorptive state. Okay, so let's talk just a little bit about liver. Liver is going to be a major site of amino acid catabolism. And it's going to take most of those carbons and make them intermediates of TCA cycle or the glycolic pathway, or to make ketone bodies. The liver can pick up both glutamine and alanine, and convert them to their nitrogens to urea for disposal. The liver also has a very high amino acid requirement. It's going to synthesize most or a lot of your plasma proteins. And these are like clotting factors, immune factors, such as your complement. It's going to make serum albumin, which is important for moving things around. It's also going to make transferrin, which is part of the clotting cascade, as well as other clotting factors. It's a major site of the synthesis of our non-essential amino acids. So it's going to take those glucose glycolysis precursors to make serine, glycine, cysteine. It's going to be able to make alanine from pyruvate. It's going to take TCA cycloimmediates, etc. So it can make a lot of the non-essential amino acids. It also has the need for disposal of xenobiotics. So it has to dispose of And remember, xenobiotics is simply just something that your body doesn't need or can't use. And so it has to do this through conjugation. And it usually does this with glycine. And another thing that it has to do, it has to synthesize heme. And it synthesizes purine nucleotides and glutathione as well. So your liver needs a lot of amino acids in order to perform its functions. Now, what about the brain? So amino acids are definitely going to be important for the formation of neurotransmitters. There are over 40 compounds or neurotransmitters that are going to need nitrogens from amino acids. So your brain needs a constant supply of amino acids. Most of your neurotransmitters are going to be formed in the presynaptic terminals, and then they're going to be stored in vesicles. And there's going to be a rapid metabolism or turnover of neurotransmitters, which requires a continuous precursor pool. The amino acids can themselves be neurotransmitters. Now, we've already talked about glutamate, which is exciting, excitatory. There's also glycine, which is inhibitory. and it's inhibitory in the central nervous system. Glycine will activate receptors, and this will result in chloride entering into the neuron. So you have this is glutamate. So glycine will activate receptors, so we'll have a neuron with a receptor, and then glycine will activate the receptor, and this will result in chloride entering. This is 1. 2, you get chloride enters. And then chloride enters through ionotropic receptors, and this causes inhibitory postsynaptic potential. All right, so the brain, of course, needs amino acid pools. So we also have different transmitters that are derived from our amino acids. So we're showing here, we have tyrosine, which is going to eventually form dopamine, and dopamine is going to be a precursor for norepinephrine. We also have serotonin is derived from tryptophan. And as I said before, our catecholamines are going to be derived from tyrosine. We also have GABA. So GABA is gamma amino butyric acid. My GABA, there's GABA. So GABA is gamma amino butyric acid. So that stands for GABA. This is going to be derived from glutamate. So this is derived from glutamate, and of course, GABA is going to be inhibitory, and it's responsible, or it's important for muscle tone. Now there's also acetylcholine, shown here. Acetylcholine is derived from choline, which is synthesized in the liver, and it also needs acetyl-CoA. Right, so those together will make acetylcholine. Now let's go back to glutamine. We talked about this once before, about glutamine and its importance in the brain. But glutamine is really a nitrogen transporter, and it helps us, which is going to contribute nitrogens for the synthesis of many neurotransmitters. Oops. So different neurons can convert glutamine to GABA or glutamate, for example, and glutamine transports excess nitrogen back into the blood. So you can see here you have in your blood your branched chain amino acids, which your blood can pick up, and the blood is going to use those branched chain amino acids for sources of fuels or carbon sources, and they're going to transfer the nitrogens to glutamate. Glutamate is going to then form glutamine through glutamine synthesis, and your glutamine can move back into another cell in the brain that might need nitrogen. So, example, your neurons can pick up glutamine. Oops. Go here. Grab that nitrogen source and form GABA, or they can form glutamate. Glutamine can also be brought back into the blood and be released into the blood, and there it can go to the gut, or it can go to the kidney, or wherever it needs to go from there. So glutamine has the role in the brain of moving nitrogen around, and it can move it outside of the brain. The brain does not need any nitrogen, it has too much, or it can move it into cells that are going to be making different neurotransmitters. Okay, so you're going to have changes in amino acid metabolism based on your diet and physiologic state. So if you have a high protein meal, here's your protein coming in, the gut and the liver are going to use most of the eaten amino acid. Right? Glutamate and aspartate are favorite fuels for the gut, and very little of glutamate and aspartate will actually enter the portal vein. Oops. Okay, so the gut is going to use these two amino acids primarily. Now the liver is going to take up 60 to 70% of the glutamate that's released and aspartate that's released into the blood. Where's my liver? Oh, here it is. And then they're going to convert this primarily to glucose via the gluconeogenic pathway. So both of these are going to go towards gluconeogenesis. And then your pancreas, don't have our pancreas. Your pancreas after a high protein meal is going to release glucagon above fasting levels, and this is going to increase your liver gluconeogenesis. Insulin is also going to be released, but it's not going to be at levels as high as a high carbohydrate meal. And remember that the amount of insulin is going to dictate whether or not your liver is itself as an organ in a fed or fasting mentality. And in a high protein meal, you have the increase of glucagon, very slight increase of insulin, and that insulin is not going to tell your liver that you're in a fed state because there's just not enough of it. However, insulin levels are going to be high enough so that your skeletal muscles are going to uptake amino acid. So we'll have our pancreas over here. Oops. I'm going to say insulin is going to be low levels. And insulin here will stimulate the uptake of your amino acids. And alternatively here, at the same protein meal, glucagon is going to keep your liver in the fasting state. So the fasting state will still keep gluconeogenesis going. Your peripheral non-hepatic tissue will also use amino acids from the diet, and typically this is going to be for protein synthesis. Okay, so high protein diets. Atkins diet, which is essentially what is now called keto diet, is where you're going to try to keep what I just talked about in play. You want to have very low insulin levels. You want your body to be taking in your fats. So you want your adipose tissue to be releasing fats. So your liver keeps using fats for energy sources instead of sugars. And you want to keep your low carbohydrates because you don't want to get those spikes of insulin and then go into an absorptive stage where you're going to build more fat. So you're trying to keep your insulin glucagon ratio at a point where you're mobilizing energy even while you're eating instead of storing energy. So this lack of energy storage coupled with loss of fat will lead to weight loss. Um, and so that's the whole premise behind those types of diets. Okay, so that is the end of this. So here's your summary. So we have to have an amino acid pool. Every cell needs amino acids to build proteins if they have that ability. Red blood cells do not. And so the cells that make proteins have the ability to break down proteins and release nitrogens. And nitrogens have to be dealt with. They are important in periods of stress because they can help to buffer acidosis, periods of acidosis. And primarily your liver will deal with excess nitrogen and formation of urea. You have a overnight fast period in which you're going to be tapping into primarily those branched chain amino acids because branched chain amino acids are a large portion of proteins. Your skeletal muscles will be using branched chain amino acid oxidation as a primary source of fuel and then releasing nitrogen as glutamine or alanine.